Mitosis by Tina, Amelia, and Alex.
Cell cycle. The cell cycle begins with interphase, which is made up of G1, S, and G2 phases. During interphase, the cell functions and grows and spends the majority of the time in interphase. After the G2 phase, mitosis occurs, otherwise known as the M phase. Mitosis is broken down into 4 parts: prophase, metaphase, anaphase, and telophase.
Interphase. Before mitosis begins, interphase occurs. Interphase can be broken into 3 phases: G1, S, and G2 phases. During interphase, the cell functions and does everything that makes the cell helpful. During G1 and G2 phases, the cell grows and develops. In S phase, DNA is replicated and synthesized.
Prophase. Prophase can be split up into early and late prophase. During early prophase, the DNA begins to condense into chromosomes. The nuclear membrane begins to break down and centrosome structures move to opposite ends of the nucleus. Also, spindle fibers begin to form in the centrosome.
During late prophase, DNA fully condenses, the nuclear membrane disappears, and each chromosome attaches to 2 spindle fibers, one on each side.
Metaphase. During metaphase, spindle fibers position chromosomes right through the center of the cell. Chromosomes are aligned down the center of the cell to make sure all the chromosomes are attached correctly.
Anaphase. During anaphase, spindle fibers are shortened and the spindle fibers pull chromatids apart towards the opposite poles.
Telophase. During telophase, chromatids reach opposite poles, the DNA decondenses, the nuclear membrane reforms, and spindle fibers disappear. Telophase is the opposite of prophase.
Cytokinesis. During cytokinesis, the ring of protein fibers separate and 2 sister cells are formed. Interphase comes and repeats afterwards.
Cell cycle control. The cell cycle is controlled by checkpoints. If checkpoints aren't completed, then the cell cycle will stop. The cell can fix the problem by eliminating itself through apoptosis or cellular suicide. An important checkpoint is R point, which occurs between G1 and S phase. R point makes sure the DNA is intact. Between G2 and M phase, the checkpoint makes sure the DNA is fully replicated. Between metaphase and anaphase, chromosomes are checked to make sure they are aligned down the middle of the cell. Without these checkpoints, mutated cells can form and can cause diseases such as cancer.
Checkpoints. Checkpoints are controlled by the activity of CDK, cyclin-dependent kinesis. CDK proteins are always in the cell but are not always active. The cyclin cycle, pictured, occurs during the cell cycle. The growth factor stimulates the cyclin process, but cyclin is put together and added to the CDK proteins when the cell begins to divide.
Errors in mitosis. PRB is the major control protein in the cell. The protein P53 regulates the PRB activity. If the DNA is damaged, the P53 protein is noticed and the P53 levels rises. When P53 levels rise, it stops the DNA replication at checkpoints. If P53 is damaged, DNA replication can occur and create benign or dangerous genes. 2 cancer-related genes can be created because of this: oncogenes and tumor suppressors. Oncogenes speed up the cell cycle and can activate mutations, while tumor suppressors can slow down the cell cycle.
This is a comparison of how CDK and PRB work together based on whether or not DNA being copied is damaged or not. The prevention of damaged DNA replication is a safety mechanism to prevent mutations.
Role of mitosis. Reproduction. Cells divide using mitosis, resulting in 2 cells that are exact copies of each other. Repair. Organisms repair themselves to repair wounds and injuries. Growth. Organisms grow and develop, making more cells.
And that was our summary of mitosis.